Thanks, Tim. Can everyone hear me okay up in the cheap seats? Okay. All right, well, thank you to all of you for having me. I'm thrilled to be here today. I'm going to tell you about data that came from a gargantuan field and lab effort and includes data from some of the most remote locations on Earth, so I don't want you to think that I did this all alone. This work is the result of joint efforts with lots of collaborators at Stanford, Scripps, UC Santa Barbara, other institutions and NGOs. As well, I want to make sure that I acknowledge all of the undergraduate field and lab assistants who've contributed to this data collection. And many of these students were at my side for multiple field seasons in the Line Islands or at the dissection table. And some even went on to do independent research projects connected with the work that I'm going to tell you about today. And I want to draw special attention to the Michigan students who've also contributed to these data in the past year and a half. Hannah, Austin, and Cassie are Europe students who've been working with me since I arrived here last fall. And then Morgan and Alexis took my Ecology of Human Parasites course and fell so hard for parasites that they signed on to do a meta-analysis project with my group. So as I talk about all this research, please bear in mind that these guys do all the hard work. Okay, so today I want to tell you about my interest in the ghosts of oceans past, the data that we can use to reconstruct marine communities as they were before human impacts, and specifically as they were before industrialized fishing changed the face of the oceans. So I'm going to start with a really brief overview of the theoretical background that motivates my work, and then I'll spend the bulk of my 45 minutes today talking about 3 research projects. One that's complete, one that's ongoing, and another that's planned. The first is some work in the Line Islands, which empirically tests the effects of fishing on parasitism. The next is a meta-analysis looking for generality in the responses of parasites to fishing pressure across marine ecosystems. And the final project is one that I would do were I offered the position here at UM. And it leverages the Museum of Zoology's fish collection in a dynamic and biodiversity-focused study that I think is going to change the way that we think about oceans past. I'll wrap up just by drawing some conclusions about how easily we can blend research, teaching, and dynamic collections-based work and use that to better understand fish biodiversity, parasite biodiversity, and how they feed back on one another. So we'll begin at the beginning. I got started in on this line of research because there's one question that really drives my interest in marine biology. What did the oceans look like before fishing? Many of us spend time on the water. If you do, you might have a sense that things are different than they used to be, and that isn't just nostalgia. There are hard data to back that up. And fishing is primarily to blame. One of the easiest ways to see how fishing reshapes fish assemblages is to think about what a trophy fish was just a generation ago. So these photos are all from Key West, same hanging board, same wharf, people fishing on the same reef. If you were a fisherman going out in 1957, you could expect to come home at the end of the day and pose next to a big grouper or even a big hammerhead shark. That was your trophy. Today, you're proud to stand next to a small reef shark or a small snapper. And there are a number of patterns that we can pick out in these photos, right? Declines in fish density, declines in fish size, and declines in the mean trophic level of fishes that are getting caught. And this is just one particularly visual example, but this is a global phenomenon. So here's a map of cumulative human impacts at the global scale. This was put together by a number of fisheries and conservation biology folks, including my PhD advisor, Theo McKelly. And here, hotter colors are indicating stronger impacts. There are a whole variety of impacts layered onto this map. But the primary impact is fishing, and it affects every corner of the global oceans. There's barely an inch that is untouched. Historical work gives us a pretty good idea of what the oceans used to look like for bony fishes and elasmobranchs. We have pretty decent data on those groups, but as many of you know, my interest in fish goes one level deeper. Each fish is an ecosystem in itself, and that internal ecosystem is typically concealed. But what if we could unveil it? What sorts of parasites would we see in this scene? Were there more parasites in an unfished ocean, or fewer, or maybe the same overall number, but a different mixture of species? You might think, who cares how many parasites there were in an unfished ocean, right? We've got so many other conservation priorities to worry about, including lots of important, charismatic vertebrates that mean a lot to us economically. But parasites are intimately connected with fish biodiversity. If fishing does cause change in parasite abundance, that might feed back to affect these vertebrates that we are very concerned about. And so here we have a big unanswered question in marine biology. And one of the things that makes this question really interesting to me is that we can posit opposite but equally reasonable hypotheses for how fishing will change parasite assemblages. So here we're going to use the same time series of dayboat trophies from Key West, and we'll layer on some dots that represent fish parasites. And this conceptual diagram will illustrate what's happening in these photos. So as we go down the photos, time is passing. As we go across the x-axis, we're getting increasing fishing pressure. Now we know that fishing doesn't reduce the density of every single fish species. Fishermen only like to pose with large, high trophic level fishes if they can get them, right? And so when those predators are taken out of ocean ecosystems, their prey are then free of predation pressure, and sometimes this can result in compensatory increases in the density of those lower trophic level species. And if that were the case, we would expect that increasing host density to result in increasing parasite transmission and potentially increased parasite abundance. And that would lead us to the prediction that perhaps fishing is leading to higher densities of parasites. In the disease ecology literature, we would call this the dilution effect hypothesis. But parasites may also become less abundant as fishing proceeds because fishing reduces fish density, fish size, trophic complexity, these things that parasites depend on for transmission. And in that case, we might expect parasite abundance to decline over time and with mounting fishing pressure. And we can call this hypothesis the healthy ecosystems are rich in parasites hypothesis after a very nice tree paper by Pete Hudson. But parasites are diverse. There's no reason for us to expect them all to do the exact same thing. And so we might also anticipate a diversity of responses among parasites in an increasingly fished ocean. And here I've changed the colors of the dots and the lines to correspond with one another. And then to me, the question really is, how does fishing affect whole parasite assemblages? Which parasites win, consistent with the dilution effect? Which ones lose, consistent with the healthy ecosystems are rich in parasites hypothesis? And then can we use some information to predict which outcome we're likely to get? This isn't a question only of interest to marine folks. Disease ecologists are also very concerned about environmental change and its effects on the transmission of wildlife and human diseases. I believe that this marine work has major relevance for broader questions that transcend ecosystem boundaries, go beyond the ocean. Marine ecosystems can help us understand these more general phenomenon because they make really great models for this problem. They contain a very diverse parasite fauna, and that's important if we're going to screen for effects across a whole variety of disease agents. These parasites are also relatively straightforward to collect, identify, and count. We already know that fishing is strongly affecting marine communities, and it changes those communities in a way that's somewhat analogous with what's happening in terrestrial ecosystems. So this is a plot from a recent science paper by a colleague of mine, Rodolfo Derzo. And these are histograms that show the frequency distribution of body mass for animals in a couple separate categories. So the first one is animals that went extinct in the Pleistocene. Those are the red bars. And then the orange ones are animals that went extinct in the Anthropocene, then Anthropocene threatened, and green is finally Anthropocene non-threatened. And the thing to take away here is that biodiversity loss hits big species first, leaving the smaller, lower trophic level species behind, potentially to experience compensatory increases in their density. And so there are some really nice parallels between terrestrial and marine ecosystems here, with biodiversity loss happening in some similar patterns. The final really great thing about fishing as a kind of biodiversity loss that we can study is that it often happens in a patchwork, right? So in marine ecosystems, we often get these areas that are off limits to fishing, either legally or because they're just impossible to get to. And so we can compare these areas and find natural experiments in which we have regions that are experiencing many of the same abiotic and biotic conditions, except they differ in the level of fishing pressure that they experience. So natural experiments are something that are really easy to set up in marine ecosystems. And we located our natural experiment in the Line Islands. So this is an archipelago of islands that contains both, there we go, inhabited and fished islands, as well as islands that are uninhabited and completely unfished. I put this picture up here so you can see how hideous this place is to work. Okay, so box goes around Hawaii, about 1,000 miles south of Hawaii are the Line Islands. And this is a color code that I'm going to use throughout the talk. Blue indicates unfished and brown indicates fished. And then the other thing that you can note on this map is that we've got 3 fished islands in the middle and then 3 unfished islands, and they are on either end of this archipelago. And This spans a gradient in productivity and probably other abiotic conditions. But because of the mixed distribution of fished and unfished islands, we can parse these potential latitudinally correlated confounders from the effects of fishing pressure. I mention this now because productivity is important later, so bear in mind that we're dealing with some latitudinal effects here. The fished islands are Terena, Tabuaran, and Christmas. They are part of the Republic of Kiribati, and each is inhabited by between 1,000 and 8,000 Kiribati people, and most of them are subsistence fishermen. So you wouldn't think that such a low level of subsistence fishing pressure by a relatively small human population would have a strong effect on these marine fish communities, but they really do. We see very few of the top predators favored by fishermen on these fished islands. In contrast, our unfished islands are Jarvis, Kingman, and Palmyra. These islands are all owned by the U.S. They're governed as they're governed as U.S. minor outlying islands, and they're part of the U.S. Pacific Remote Islands Marine National Monument. And that designation involves really strict bans on fishing that are well enforced. As a result of those protections, these islands are teeming with predators: sharks, jacks, snappers, groupers. They're truly a window into what coral reef ecosystems used to look like prior to the onset of fishing pressure. So we set out to understand how fishing can drive change in parasite assemblages by conducting sampling across those 6 islands. The sampling was done on a National Geographic Society cruise that was run in the fall of 2010 by my collaborator Stuart Sandin and his student Brian Zaglinski, both at Scripps. And these guys sailed from Oahu all the way down the Line Islands chain. At each of those islands, they shot fish with pole spears. And we focused on 7 host species for this part of the study. We chose these species to span a variety of trophic levels and body sizes so that we'd maximize the parasite diversity that we detected in them. And overall, we dissected 945 fish for the project across 7 host species and 6 islands for a total of 44 parasite species, just shy of 1 million individual parasites. And this is what those dissections look like. We pull out all the visceral organs, we isolate all of the other organs that we're interested in, and then each one gets examined individually under magnification for metazoan parasites. We do not look for protozoa, viruses, or bacteria. And we'll begin the results by taking a look at parasite diversity. Now here on this plot, I've got our 3 unfished islands on the top of the y-axis, and the 3 fished islands are on the bottom. The x-axis is a jackknife estimate of parasite taxon richness, so you can consider this to be parasite richness corrected for differences in sampling effort. And I should also mention that this and all the following analyses are going to control for potentially influential covariates. So that includes host body size, the depth at which the host was collected, latitude of the island, and then random effects for host species and island. And here's where our mean effects fall out for each of the 6 islands with 95% confidence intervals. And we can also draw grand means for unfished and fished islands. So those shaded areas correspond to the 95% confidence intervals for the grand means. And what we see is that richness is about 50% higher on the unfished islands than on the fished islands. So essentially there are many more kinds of parasites on the unfished islands. Not necessarily more parasites, but more kinds of parasites. So already we see a way in which fishing might drive change in parasite assemblages by eroding the species richness of those assemblages. But we're also very interested in knowing parasite abundance and how those patterns differ between fished and unfished islands, so that will form the bulk of the rest of this results section. Okay, so I'm going to orient you to the results for abundance by starting with one panel of what will eventually be a multi-panel plot. So this one corresponds to one of our 7 host species, Tinochaetus marginatus. That's a bristletooth. On the y-axis, you'll find all of the parasite taxa that we detected in this particular host species. For the parasite fanatics in the audience, the alternating shaded and light bars indicate groups of parasites, so crustaceans, monogenes, trematodes, cestodes, nematodes, and acanthocephalans at the bottom. On the x-axis is a standardized regression coefficient for the effect of fishing. So that's a coefficient that comes out of the generalized linear mixed model that you see at the bottom of the slide. And there's one of those for each parasite taxon we found. So basically, positive values on this x-axis indicate a positive response to fishing pressure, or parasite taxa where we found more individuals on unfished than on fished islands. And then negative values indicate a negative response to fishing pressure, or parasite taxa where we found more individuals on unfished than on fished islands. And because this is a standardized regression coefficient, anything greater than +2 or less than -2 is significant. That's why those areas are shaded. And finally, all the p-values that you see on this plot are corrected for multiple comparisons. So with all of this in mind, we can take a step back from this plot and appreciate that what it's telling us is that there are several parasites that don't care whether fishing is happening or not. They are equally abundant on fished versus unfished islands. And those are the parasites that you see in the white band in the middle. There are 3 parasites that have a significant positive response to fishing pressure, and that's consistent with the dilution effect. And then there are 3 parasites that have the opposite response. They actually decline in the presence of fishing, consistent with the healthy ecosystems are rich in parasites hypothesis. And then if we zoom out and take a look at all 7 host species, we see that the same pattern holds. Many parasites don't respond, some respond positively, and some respond negatively. And these data suggest kind of a subtle change in parasite assemblages as fishing proceeds. Not an all-out increase or decline, but a reduction in diversity and a shift in composition. We've got some parasites that do what the dilution effect hypothesis predicts. We've got others that do what the healthy ecosystems are rich in parasites hypothesis predicts. And that's really interesting, right? We've got this kind of variability, and we could have stopped there, but we had some hypotheses about why there might be this kind of variability, and we thought that it might have something to do with parasite traits. So we sorted our parasites into 2 broad types of transmission strategies, directly transmitted and trophically transmitted. Directly transmitted parasites move from one host to another host of the same species. Trophically transmitted parasites move from one host to another host of a different species, usually in a sequence. We found parasites in both of these groups, crustaceans and monogenes among our directly transmitted parasites, and nematodes, cestodes, trematodes, and acanthocephalans among our trophically transmitted parasites. And we made some predictions based on these fundamental life history differences. Assuming that these parasites would be transmitted in a density-dependent manner, we predicted that we might reduce the density of the hosts of directly transmitted parasites and see a corresponding reduction in the transmission of those parasites and in their abundance. But we know that reductions in abundance are not the only thing that we can see among fish species exposed to fishing pressure. We might also see increases in host density if, for example, we remove the predator of these hosts by fishing and release them from predation pressure. And in that case, we would expect to see an increase in transmission of their parasites. So we predicted that directly transmitted parasites would track their host's abundance. In contrast, trophically transmitted parasites are dependent on multiple host species for the completion of their life cycles. And those life cycles often include the large, high trophic level fishes that fishermen prefer. And we know if we lose one link in the life cycle of a complex life cycle parasite, it's game over. Transmission stops for that parasite. So we expected trophically transmitted parasites to be especially sensitive to the impacts of fishing on their hosts. So to test these hypotheses, we wanted to turn this analysis on its head. We essentially wanted to group up these regression coefficients by parasite type and see if there were any systematic patterns. And since we wanted to be able to incorporate the error associated with each one of these coefficients, we used the statistical techniques of meta-analysis. This isn't a true meta-analysis because the data come from our own data sets and not from the literature, but we used the statistical techniques of meta-analysis. Okay, so here we're going to break down the mean effect sizes, which just like before, positive response indicate a positive response to fishing pressure and negative values are a negative response to fishing pressure. We're going to break that down by higher order parasite taxon. So we've got crustaceans and monogenes up top, and then trematodes, cestodes, nematodes, and acanthocephalans. And those are the trophically transmitted parasites at the bottom of the plot. And here we're going to plot mean effect sizes again with 95% confidence intervals. If we begin at the top, we see that the crustaceans and the monogenes both have significant positive responses to fishing pressure. They're both more abundant on fished islands than on unfished islands. And then if we combine them up as our only 2 groups of directly transmitted parasites, we see that that result remains. So why is there this positive response? We predicted that directly transmitted parasites would track their hosts, and it turns out that's exactly what they're doing. So here on the x-axis, we have the host's response to fishing. So negative values on this axis indicate that the host is declining in density. So that might happen if, for example, the host is fished. And then positive values on this axis indicate that the host is responding positively to fishing pressure, so perhaps experiencing compensatory increases in response to release from predation. And then there's parasite response to fishing pressure on the y-axis, same interpretation there. And what we see is that the directly transmitted parasites have a positive relationship, so parasites of increasing hosts increase. And that makes sense. Directly transmitted parasites are sensitive to changes in their host density because there are no other hosts in the life cycle for them to worry about. Unlike trophically transmitted parasites, their transmission can be dependent on the density of just one host species. And as it turns out, we had a number of hosts that increased in abundance in response to fishing pressure. Now, this is not a perfect one-to-one relationship. There are several parasites that have a positive response to fishing pressure, despite the fact that their host is pretty much not responding to fishing. And this might be because these parasites are not specific to a single host species, and perhaps they're responding to increases in the density of other host species within their host range. But in general, we see that directly transmitted parasites are more abundant on fished islands if their hosts are more abundant there. So that's what's going on with the directly transmitted parasites up top. If we move down to the trophically transmitted parasites, it's a little bit of a different story. The cestodes and the nematodes both have significant negative responses to fishing pressure. They're more abundant on the unfished islands relative to the fished islands. And if we only found this one acanthocephalin species, so it wasn't included in the meta-analysis, but I did want to plot it for you here so that you could see that it is non-significantly negative. And so far, this is all consistent with our hypothesis that fishing should interrupt the life cycles of trophically transmitted parasites. But the trematodes don't conform to that pattern. They don't differ significantly from zero, unlike the rest of the trophically transmitted parasites, and we'll talk in just a second about why I think that might be. But if we average over all of these trophically transmitted parasites, we see that their response to fishing doesn't differ significantly from zero, but it is significantly less positive than the response of the directly transmitted parasites. Okay, so what's going on with the trematodes? I think it has something to do with their life cycles. They differ a little bit from the remainder of the trophically transmitted parasites. Cestodes, nematodes, and acanthocephalins start their lives as eggs in the intestine of large predatory vertebrates. Those eggs are expelled with the feces out into the environment, where they then hatch and infect a crustacean first intermediate host, often a planktonic copepod like the one shown here on this plot. The trematodes are similar. They also use large predatory vertebrates as their definitive hosts, but they tend not to use elasmobranchs. They prefer bony fishes. And their first intermediate hosts are snails, not crustaceans. Now, planktonic crustaceans are pretty insensitive to fishing impacts. Their abundance is determined by bottom-up effects like productivity. So when fishing hits the life cycle of a cestode, nematode, or acanthocephalin, the effect is straightforward. It's a reduction in the final host and a reduction in transmission. We also may see that reduction in the final host for the trematodes, but we know from work on other coral reefs that snails can sometimes experience strong compensatory increases in abundance in response to fishing because they are often the prey of fished species. So I want to emphasize that we don't know this from this particular system, but it's been shown robustly on other coral reefs that this can happen. So trematodes might get these kind of counteractive effects, right? A loss of their definitive hosts and an increase in their first intermediate hosts. There is one example that suggests to me that this is what's happening, and I want to show it to you here. We had this one genus of trematodes called Stephanostomum that we detected in 5 host species that we dissected. And like I said before, trematodes usually use snails as their first intermediate host. Stephanostomum uses big, meaty moon snails and whelks as its first intermediate host, and it's a trematode that has an exceptionally positive response to fishing pressure. So I think that this is suggestive that Stephanostomum experiences an increase in its first intermediate host in the response, in response to fishing pressure, and that this might explain part of why the trematodes have this equivocal response. But if we look over all the trophically transmitted parasites that lack snail intermediate hosts, we see that they do have a significant negative response to fishing pressure. So this data set shows us that there are winners and losers among parasites exposed to fishing pressure. In the system, the losers are trophically transmitted parasites, especially the cestodes and the nematodes, and we see that overall parasite diversity takes a big hit in the presence of fishing. The winners are directly transmitted parasites of hosts that increase with fishing, and then trematodes with intermediate hosts that increase with fishing. And we were really excited by this finding. You know, it suggests that oceans past may have been dominated by a completely different set of parasites than we see today. Overall levels of parasitism haven't necessarily changed, but parasite species composition may have been remade. This across-island study has the benefit of letting us empirically test the impacts of fishing at a really big spatial scale. So then we set out to see if they would hold at a finer resolution. We did some in-depth sampling on a single island. We chose to work on Christmas. Here's a map of Christmas Island. It's one of the inhabited line islands, and here everyone lives on the western side, and since it's most convenient, that is also where they fish. And these circles, the blue circles, show you relative fish catch and that was taken from a household survey, and that extrapolation made for each individual site where we were sampling fishes. And so that survey shows you that there's lots of fishing on the western side and very little fishing happening on the eastern side, which sets up a really nice gradient for testing the effects of fishing pressure on parasite assemblages, but there is a catch. Productivity is confounded with fishing pressure across this gradient. So here the squares indicate concentrations of chlorophyll A, with hotter colors being higher concentrations. And on this particular island, the current sweep from east to west, which creates this effect called island wake upwelling on the western side of the atoll, which is why you see that increase in the concentration of chlorophyll A, increase in productivity. And it's really difficult to disentangle this productivity gradient from the fishing pressure gradient. They are very much bound up with one another. We did find a way to use this gradient to our advantage, which I'll explain in just a minute. But we spent about 2 months on Christmas in the spring of 2010 collecting fish across that fishing pressure gradient using the same protocols as we did for the across islands analysis. And in total, we dissected 894 fish for this project across 5 host species and 25 sites for a total of 47 parasite species and a little bit less than 69,000 individual parasites. And I want to point out here that this data set is completely independent of the across islands data set. They are totally orthogonal. Okay, so the analysis had multiple steps, and I want to walk you through it. Basically, we did the whole thing exactly the same as in the cross-island study, but instead of using this model, we used this one. So instead of trying to parse fishing status and productivity apart from one another, which it was possible to do in the across-islands gradient, we modeled them as one variable for the within-islands analysis. We didn't try to pull them apart at all. And we asked, how do parasites vary across this combined fishing pressure and productivity gradient? Then we took the regression coefficients from that analysis and we meta-analyzed them to get mean effect sizes for the big parasite groups, crustaceans, monogenes, trematodes, cestodes, nematodes. And then we fed these mean effect sizes into a simple regression where we asked, are the results from the combined fishing and productivity gradient within islands consistent with the mean effect sizes for fishing and mean effect size for productivity that we found in the across islands gradient. So basically we used the information from the unconfounded cross islands gradient to parse the relative effects of productivity and fishing pressure for the within island gradient and to ask, do these results from 2 entirely different spatial scales agree with one another? And what we found is that they do. So here are the mean effect sizes for the across islands analysis with negative responses on the blue end of the heat ramp and positive responses on the brown end. And each one of these corresponds to one of the big parasite groups, nematodes, cestodes, monogenes, trematodes, and crustaceans. And then these are the residuals for the effect size of fishing in the within islands gradient. So basically the variability that's left after we remove the effect of productivity. The within island results overlap quite nicely. The agreement between the 2 data sets is statistically significant, and despite the fact that we are at radically different spatial scales in these 2 data sets, we see similar patterns. Just like before, fishing causes declines in trophically transmitted parasites and increases in directly transmitted parasites. So what we're seeing here is that the oceans may have looked different in the past than they do today, right? This is not necessarily an overall increase or decrease in disease, but a shift in the kinds of parasites that are common, a reduction in parasite diversity and a shift in the composition of parasite communities. That result is really exciting. It's unexpected. It leads to some really interesting follow-on questions, but I wondered something. How general are they? Is this a phenomenon that we see at this one quirky little archipelago of islands and that won't be repeated anywhere else on Earth? To answer that question, my collaborator Kevin Lafferty and I performed a global meta-analysis of published studies which compared reserves and open access areas in terms of their parasite assemblages. We did not include the results from the Line Islands because we wanted to see whether the existing data in the literature would agree with our comprehensive data set of parasites from the Line Islands. We extracted 74 host parasite combinations altogether, and these came not just from coral reef ecosystems, but from lots of other marine ecosystems as well. And we essentially pooled replication across those combinations to see if we could pull out generalities. What predicts when parasites increase in response to fishing pressure and what predicts when they decline? Okay, so here on the x-axis we've got the mean effect size, which just like before is the parasite's response to fishing pressure. So positive responses and negative responses. On the y-axis we break things down as all parasites combined, directly transmitted parasites, and then trophically transmitted parasites. And then if you look in the legend up at the top of the slide, you'll see that we are actually able to get even a little bit more nuanced with this data set. We're able to distinguish parasites with a fished host at some point in the life cycle, and that's the brown triangles, and parasites with no fished hosts at any point in the life cycle, blue circles. And what we see is just like in the Line Islands, the trophically transmitted parasites decline and the directly transmitted parasites increase. If we then break things down a little bit further, within the directly transmitted parasites, there is no difference between those that use a fished host and those that use an unfished host, but that overall their response is positive. So this might be because directly transmitted parasites have broader host ranges. Maybe they're able to switch from a host that's declining to a host that's not declining, or even a host that's experiencing compensatory increases in density in response to fishing pressure. But one way or another, the fishing status of the host does not matter for these directly transmitted parasites. They have, in general, a positive response. The trophically transmitted parasites are, again, a different story. The fishing status of the host is very important for these guys. So we'll start with this brown triangle on the left. These are trophically transmitted parasites with a fished host at some point in their life cycle, at least one fished host. And we see that they have a strong negative response to fishing pressure, which is exactly what we would have anticipated and exactly what we saw in the Line Islands data set. But then we also see that trophically transmitted parasites with no fished hosts at any point in the life cycle have a strong positive response to fishing pressure. So what's happening here? I think it's possible that trophically transmitted parasites with no fished host at any point in the life cycle are merely more likely to have some fished host that increases in density in response to fishing pressure. And this would be somewhat analogous to the trematode example from the Line Island study. So fishing status of the host appears to be really important for these trophically transmitted parasites. And overall, just like we saw in the Line Islands, there are winners and losers among parasites exposed to fishing pressure. The losers are trophically transmitted parasites, the winners are directly transmitted parasites, and those trophically transmitted parasites with hosts that increase with fishing pressure. And what we have here are strong, consistent patterns from a variety of marine ecosystems that show that human disturbances like fishing can increase or decrease parasitism depending on the parasite's identity and the host's fishing status. So this confirms that fishing can drive these compositional shifts in parasite assemblages away from complex life cycle parasites and toward directly transmitted parasites. And now all that's well and good, but I'm not stopping there. My work has been the first convincing demonstration that the differences in fish assemblages that we see in fished versus unfished environments echo in the parasite communities of those environments. Fish biodiversity and parasite biodiversity are intertwined in what affects one affects the other. And the Line Islands were a really great system for doing these initial studies, but they constrain us in some ways. There's strong contrast between the fished and unfished islands, and with only 6 islands in the chain, they only give us this static contrast, fished versus unfished, 2 ends of a spectrum. So it kind of looks like this. Along the x-axis, let's say that we have increasing fishing impact. Along the y-axis, we've got some metric of parasite assemblage composition. And then in the upper left, we've got Palmyra and our unfished islands, which are pristine and have some sort of parasite assemblages that characterize them. Along at the lower right-hand corner of this plot, we have Christmas and the other islands that are impacted with some other kind of parasite assemblage. And we've only got the ends of the spectrum, right? We can only draw the outer limits on this relationship. But what if we wanted to understand the functional shape of that relationship? Do we get a one-for-one change as we shift the fishing status of an island and look at its parasite assemblage, or can we fish really hard before we see any change in parasite assemblages? Or does a small fishing impact result in a large change in the parasite assemblage? We can't look for these kinds of tipping points in the existing data set. 6 islands are just not enough. But we can if we expand our scope. So that's what I'm doing right now. I'm currently continuing my work on these coral reef fish parasites. We're adding 5 additional islands, and those 5 islands have diverse histories of fishing pressure. So they're the yellow ones that you see at the southern end of this archipelago. They're all part of the same archipelago. The southern ones are south of the equator. And with 11 islands, we can now start to draw curves and ask what level of fishing pressure, what amount of change in the fish community is sufficient to change parasite assemblages, and what do those changes look like? I've already begun work to answer these questions. I currently have a freezer full of fish, and more are coming soon. And so far, we've handled a number of fish specimens collected from these 5 islands. More are en route. What I really need are graduate students who are ready to take this project and make it their own. If I were to be offered the position here, these projects are ready to go on day one. We already have the fish, the protocols, and the metadata in hand. And it would also be worthwhile to discuss whether we can contribute these fish to the Fish Division's collection, something that I'll talk about in just a couple of minutes. Okay, so I've told you about my empirical parasites that make use of spatial variation in fishing pressure to draw inferences about how fishing drives change in parasite assemblages over time. But now I want to talk about a project whose time is really long overdue, a project that attempts to reconstruct historical parasite assemblages in a novel and innovative way. So people have come up with some pretty creative means to glean information about historical assemblages of fishes. Some have used art, like this Roman mosaic that shows a dusky grouper, a species that today is endangered and very rare in the Mediterranean. Others have used the composition of fish species assemblages reflected in seafood menus over the years to understand how those assemblages have changed. And of course, people love posing next to pictures of the big fish that they catch. And creative folks have extracted from these photos either qualitative or sometimes quantitative information about what fish assemblages used to look like. And some of these photos are just so striking. They really just bring home how much the oceans have changed, even just over a couple of decades. Parasites are not that charismatic. You don't take pictures of them while you're on vacation. Hopefully they're not on your menu, and there are not that many works of art devoted to them. So historical information on parasite assemblages is much harder to come by, except for one source: museum specimens. When fish are preserved in ethanol or formalin, their parasites are preserved right along with them. And here in the museum's division of fishes, we have 3.5 million cataloged specimens with some collections dating back to the 1850s. This sets us up to be able to reconstruct parasite assemblages over a broad sweep of time. So I propose to begin building a time series of parasites in common species that are well represented over long time frames in the fish collection, and pairing fished and unfished species to really dissect how fishing impacts parasite assemblages. This is a project that I've been itching to do for a really long time. I've actually piloted it at both the California Academy of Sciences in San Francisco and the Smithsonian Institution's National Museum of Natural History in DC. And so here you see a northern anchovy from the Cal Academy that was collected in 1951. The dissections of these preserved fishes are only a little bit trickier than they would be for fresh fishes. Of course, we want to be able to return the carcass to the collection, so we do a more limited parasitological dissection, but we're still able to get at most of the parasites that we're interested in. So here's a nematode that we found in that particular anchovy species. Even though the fish are more than 60 years old, we can still detect their metazoan parasites, and in many cases identify them to a pretty low taxonomic level. And after the dissection, the visceral organs go right back into the body cavity, the fish gets stitched up, and it can be returned to the collection with most of its external morphological features pretty much preserved. And I don't want you to think that we just find nematodes. We would expect to find nematodes in preserved fishes because they have a cuticle, they probably preserve really well, but we also find really delicate parasites in these fishes. So this is a trypanorhynch tapeworm larva that I pulled out of a cod in the Smithsonian, and there were a number of these larvae just in this one cod individual. Now, this suite of projects will be the first to give us hard empirical evidence of what marine parasite assemblages looked like before the onset of industrialized fishing pressure. And we're on the leading edge of what I think is going to be a flood of research utilizing museum collections. Museum collections are becoming recognized as this superior window into the past, and I think this is particularly true in disease biology, where we often have few other options for getting historical baselines. And just last week, a really cool paper came out in Ecology by Guilherme Becker, and here, this is a preprint ahead of publication. The paper uses museum specimens to reconstruct historical baselines for Batrachochytrium dendrobatidis, BD, the chytrid amphibian-killing fungus. And Becker and his colleagues used qPCR to detect BD in Leptodactylus frogs collected over a 119-year time span in the Amazon, which is really just remarkable. So here's a map of the distribution of their negative and positive samples. I don't want to go into too much depth on this particular study, except to show that they kind of reveal an interesting, maybe unexpected pattern. That purple line is the overall prevalence. The gray bars are sample size from time bin to time bin. And the overall prevalence is relatively stagnant, although they were able to identify a cluster of cases that coincides with reports of amphibian die-offs from the 1990s. So really, really interesting stuff, information that's absolutely critical if we're going to understand and manage disease. And I think that museum specimens represent this really untapped resource for understanding the dynamics of ecosystems in response to global change. But in addition to tackling this big, important, and unanswered question, it presents an opportunity for really nice links between research and education. So this past winter semester, I taught a really successful class called Ecology of Human Parasites. Many of my students are in the room right now. And that course emphasized hands-on and inquiry-based learning. And now you will read some of the things that the students in this room right now wrote about this course in their anonymous course evaluations. The interactive labs were fun, the lab sessions were engaging. I wish it didn't have to end. Right? So the lab portion of this course was the thing that the students identified as their favorite part, the thing that they learned most from. And in an ecology and evolution of fishes course, were I to be offered the position here, I would give the students more of what they want and more of what they learn best from, which is hands-on labs. I would run labs that take advantage of the fishes in the museum's collection, including labs that have the students contribute to our ongoing data set of parasites in fishes. And that gives the students the opportunity to see the way that real science is done with all of its wrinkles and surprises. And it's totally doable. I've actually run labs like this at Stanford when I was a graduate student. And in my past teaching experiences, hands-on labs have definitely been the way to get students to absorb all of these facts about fish anatomy and physiology, ecology of fishes, ecology of fish parasites. Lots of information to take in, but it's easily assimilated if you have this kind of framework of hands-on experiences to link it to. And the more creative we can get with these activities, the better. And we can get pretty creative. So this is an ocean sunfish that I had my Stanford students dissect during one of our lab sessions. And this creature is as bizarre externally as it is internally. And the other really cool thing that I like about ocean sunfish is that they are stuffed full of parasites. So this is just some of the adult tapeworms that we pulled out of the intestine of this one poor juvenile ocean sunfish. And the students get to see these parasites, if not their host, alive. In my experience, students love getting their hands dirty, and that's exactly what they would get in my Ecology and Evolution of Fishes course. So this is a project that I think is a really ideal marriage of undergraduate teaching and cutting-edge research. It will spur active use of the museum collections, and I think that this kind of dynamic use of the collections could really elevate the visibility of the museum, both within LSA and within the broader fish ecology and disease ecology worlds. Okay, so I'll wrap up with just a couple words about teaching and research on oceans past. Parasites are a hugely diverse and ecologically influential set of symbionts for fishes, and yet we just don't even have the foggiest notion of what parasite assemblages looked like before the onset of industrial fishing pressure. And all we can do at this point is look for ghosts. I want to end by telling you a quick story that hints at the kinds of ghosts that we might see. As you well know by now, the average tapeworm has a complex life cycle. It reaches maturity in the intestine of a large vertebrate like this shark. It produces eggs, and those eggs are expelled in the host species. Enter Amphilyna. Amphilyna is a tapeworm. It doesn't look like one, and it has a really weird life cycle for a tapeworm. It has dispatched with the need for a final host. It reaches maturity and produces eggs in the body cavity of its fish second intermediate host, which in the case of Amphilyna is a sturgeon. Now, two weird things about Amphilyna. This egg-producing stage is the one that's displayed on the slide, but this does not look like an adult tapeworm. In fact, it looks like a larval tapeworm. And it's producing eggs in the body cavity of its host, which in any other fish would be a pretty dumb move, considering that the body cavity isn't typically open to the outside world, where the eggs need to go to complete their life cycle. But sturgeons have coelomoducts, which connect the body cavity to the cloaca. So what's going on with this tapeworm? Well, a lot of folks think that this foreshortened life cycle is a response to the decline of dinosaurs. That as Amphilyna was losing its final host, it struck upon an adaptation to keep on going. So Amphilyna is a ghost. It's an extremely lucky tapeworm of dinosaurs. It was probably one among many genera of tapeworms that disappeared with the decline of dinosaurs. Varsity Drive is haunted. All museum collections are by ghosts of oceans past. The empirical work that I've been doing presupposes that parasite assemblages of the past were made of the same stuff that they are today. But what if they aren't? Museum specimens are the only way forward for this kind of work, and with one of the largest fish collections in North America and a commitment to support dynamic, collections-based biodiversity research, there are not many other institutions that are as well positioned as ours to make progress on this problem. This is a whole new frontier in the study of fishes, and one that Michigan could be poised to lead. Thank you. I think we've got a couple minutes for questions. Deborah.
So you talked about one very important trait of the parasites that could affect what the impact of fishing would be on them, and that's the mode of transmission. Were you able to look at any others, or isn't enough known? So one thing I'm thinking of, are they generalists or specialists, or the one that clearly could have some impact?
Yeah.
Is enough known about them where you can start looking at effects of those?
That's an excellent question. Yes, so we tried to look at host specialization. Unfortunately, one of the downsides of working in a place like the Northern Line Islands is that the parasite fauna is actually relatively poorly described. Right? So we did our best with identifying these species, but because they're not even yet described, we can't be very sure about their levels of host specificity. That said, host specificity is relatively well conserved, at least for the vertebrate definitive hosts within these larger taxonomic groupings of parasites. And when we assigned it that way, like when we said, okay, like copepods are generally generalists and monogenes are generally specialists, we didn't see any pattern there. So it's possible that were we to do this with a more well-described parasite fauna, we might be able to find those patterns, but we didn't see them with what we know of the parasites in this group.
What if I could kind of build off of that question? It's very closely related to the thing I was wondering about. So we know that huge amounts of material and energy flow through the parasitic infections. Even if you can't resolve it to the level of species, you can still talk about functional groups using, if nothing else, the data that you've collected right there. Have you looked at the changes to that network of connections amongst functional groups or amongst species to the extent you can to see if there's any interesting differences in the network structure?
No, we haven't, but that's a great idea. Maybe we should start up a second collaboration to work on that together. Yeah, no, that's an excellent point. The studies included in that meta-analysis did not include any dynamite or cyanide fishing or any of those like really destructive fishing techniques. I think it was, so there's a little bit of commercial fishing, which is probably done with trawls, and then there was also, I think, at least one or two recreational fishing papers, which would have been hook and line, but nothing as interesting as dynamite fishing, unfortunately. Like you.
I have a very naive question. So, do parasites really contribute anything positive to the host, particularly the ones that go through this trophic transmission mode that involve multiple species in the system?
Yeah, no, that's an excellent question. And actually, Priscilla and I were just talking about that. For the complex life cycle parasites, they actually are really, really beneficial for one kind of host in their life cycle, and that's the definitive host. And that's because they can sometimes cause behavioral changes in the second intermediate host that makes it more likely to be eaten by the definitive host. So this is in the parasite's interest because it wants to complete its life cycle. It is not in the second intermediate host's interest at all. But it's thought that this is a shunt, basically, a way that prey species move into predator species where they wouldn't otherwise have done. Essentially, it's a way of making more resources available to predators, and that's entirely a parasite-mediated interaction. And I actually think that's one of the things that makes, well, to me, that's the most interesting kind of future research direction in terms of the ecological importance of parasites. I think that's a really under-recognized factor that makes them important in ecosystems.
Because the same thing happened in the plant system. So now I really suggest you should change the name from parasites to symbionts.
Symbionts. Well, so in the plant literature, aren't parasites just symbionts that have a negative effect on their?
We have enough symbionts now recognize the roles are much more important in
Yeah,
the
that's a
evolution
good
of
point.
plants and the symbionts.
And they can shift back and forth. Yeah, thank you. Oh, you mean like the latitudinal gradient?
Yeah, obviously you have your hands full, but would you believe that somewhere where there's more available oxygen in an ecosystem might have more, a larger diversity of parasites?
Yeah, it's totally possible, and and to some extent, it depends on the kind of parasite too. Like, there's this little cottage industry of parasites as bioindicators, where we're kind of piling up examples of systems in which parasites increase in response to X, Y, or Z impact. One of the things that fish parasites in particular are expected to be pretty sensitive to is oxygen, particularly those that have an environmental stage. These are little guys. They depend on having a large supply of oxygen available for them. So definitely possible, yeah.
Is there a meta-analysis referring to this?
No.
Do you know whether or not research
Could be. Definitely could be.
Question for another day.
Yeah, I think that's fertile ground for another meta-analysis.